Good day, grade 12 learners. Welcome to today's Life Sciences lesson. My name is Shabnam Suknandan. This lesson is brought to you by the Gauteng Department of Education in collaboration with the Saibono Discovery Center. So today, learners, we're going to be doing paper 2 revision that you're going to be writing on Monday, the 22nd. And remember, this revision lesson is going to be brought to you by myself and my colleague Sina Sikeke, who will be doing the second half of the presentation. So without wasting time, learners, here is a very important page which you need to take actually a picture of, right? You need to actually take your cameras now and focus on the Instagram QR code and also that of the learners' resources. In the Instagram, you're going to actually get a whole lot of things that's going to help you, like little video clips on genetics. You're going to have little video clips on the evolution of the skulls and the trends in evolution, right? A whole lot of investigative questions and how to answer it. And in your learner resource, you're going to get a lot of past papers from different provinces together with the marking guidelines. So please, without wasting time, please take your phones and actually focus onto this QR code and get it right so that you have access to all the resources in life science. Okay, remember that's the Instagram page and that is a resource folder in the Google Drive. So you have two separate pages, one with the videos and one with all the resources for grade 12, including your one pages, your sub booklets, and your voice notes as well. So if you have no teacher or you're studying on your own, then you can simply put in the voice note, which actually gives you the recording of a topic. And that's very helpful because you can sit anywhere and you can listen to a whole topic at the comfort of your leisure. Okay, so that's the Instagram page and that's the resource folder in the Google Drive. Okay, right, let's move on. Okay. So children, what's very important is that the examiners use one very important document, and that is the exam guideline. And this exam guideline tells you exactly what are the weightings of the topics and which topics you should actually focus on. Now, if you look at paper 2, it only has 4 topics compared to paper 1, which has 7 huge topics. Paper 2 has an advantage. You have to learn it over the weekend. You have a Friday, Saturday, and Sunday, and you learn over 3 days. You can learn 4 topics. So please allocate your time very wisely for you to get the maximum amount of marks. Let's see. First of all, in paper 2, it covers DNA and meiosis, which was done in term 1. Now those 2 topics, the DNA is 18% of your paper. Remember, you need 30% to pass. That 18 out of the 30% is on DNA. And that is an allocation of 27 marks, which Sina is going to be doing on. And then meiosis is going to be 14% of the paper. And that is 21 marks. So altogether, the DNA and meiosis is carrying a total of 48 marks, which is actually 32%. Now, that's a lot of marks just knowing and mastering those 2 topics. And the better part is that term 2's work and term 3's work is genetics and evolution, where genetics is occupying a whopping 48 marks. That's 32% of your paper. So if you know all the genetic crosses that we're going to be mentioning in this revision program, if you know the recipe of that, and you can get a whopping 48 marks, you are already close to getting a distinction. Right, and then evolution, on the other hand, takes 54 marks. That is 36% of your paper. So just evolution and genetics is taking a total of 68% of paper 2. So children, if you are in level 1, just 2 topics, genetics and evolution, is going to give you 68%. So if you really master those 2 topics, you can't fail life sciences. Okay, so this is the guideline for paper 2. Right, so we're going to be covering evolution, genetics, DNA, and meiosis. Now here is an in-depth focus on the scope of your topics. We're going to talk about now. I'm going to first do evolution, which is the biggest topic. So we're going to start off with that. Right? And here you have an exam guideline which your teachers must have issued to you. And if you don't have it, I told you you had a page. We can always project the page at the end if you didn't capture it, where you're going to get all your resources, including the exam guideline. And this is just basically telling you that for evolution, you need to know the 3 theories that propose evolution, the scientists who propose those 3 theories, and also exactly who are the people that propose those theories, right? And what does it entail? So here you can see Lamarck, Darwinism, and punctuated equilibrium are the 3 theories. And then Lamarck's theory, law of use and disuse, law of inheritance of acquired characteristics, and then Darwin's natural selection. Here it's very, very general. So they don't normally give you a general description in the paper to answer. They will ask you to apply the general description to a phenomenon in the paper. So they can give you a little case study, right, where you must read, and then you must now apply Darwin's theory of natural selection to that little paragraph, okay, where there's a great deal of variation amongst the offspring, and then there's some offspring that have a favorable trait. You must identify from the passage what's the favorable trait, what's the unfavorable trait, okay, and then you must always indicate that there has to be some form of selection pressure, either predators, changes in the environment, changes in the temperature, changes in the pH, some kind of an environmental pressure, okay, or competition. And then because of that, now the one with the favorable trait will have the trait to survive that change in the environment, and the one with the unfavorable will not be able to do it, okay. And then obviously those that have the favorable trait will be able to have the gene flow and pass the genes to the next offspring with a favorable trait, and a greater proportion of offspring will have that favorable trait naturally, okay. So you need to look at this, read it, and then apply it to the scenario that they give you in your paper. If they just say, write Darwin's law of natural selection, or write down Darwin's theory of natural selection for 6 marks, then you can give this general description. But if they ask how would Darwin use natural selection to this scenario that they gave you, then you must take this and apply it to the example given in the paper. Punctuated equilibrium is just at the speed at which evolution takes place, and that is done by Eldridge and Gould. Okay, so we'll look at that a little more in detail later. And then we're going to look at Lamarck's theories. Remember, we said Lamarck proposed the theory of why there is variations in the offsprings. What is evolution? It's a change in the structure and behavior of organisms over a period of time, that what exists today actually descended from an ancestral form, right? Now, this theorist or scientist called Jean-Baptiste de Lamarck proposed 2 laws. He proposed the law of use and disuse, right? And he wanted to use that law to explain why there's certain organisms that look different from what existed in the past. So he used basically a giraffe, right? So the law of use and disuse states that as an organism uses a structure or organ more regularly or frequently, it becomes better developed and even enlarged. And if that organism does not use a structure frequently, it will become less developed and reduce in size, or may even disappear completely. So here you can see that Lamarck said that the ancestral giraffe, the first giraffes, had short necks. But then there was a change in the environment. There was some kind of a selection pressure called competition. Many other herbivores were competing for the lower branches of the grass and the leaves, right? And the giraffe spotted that on the upper branches of the tall trees, no one was eating those leaves. So there was plenty leaves on the top. So the giraffe, because of the competition, it decided to stretch its neck more and more and more. And as it stretched its neck more and more to reach the top leaves, its neck became longer and remained like that. So what does it mean? It means as the giraffe used its neck more and more, the neck became longer. So that is called the law of use and disuse. Okay. Whereas in snakes, where, snakes used to have legs before, the ancestral snakes had legs, and because the legs were in the way when they had to hide away from the big predatory bird, the snakes tried to use their legs less and less and less, and that is why today snakes have no legs. So that law of disuse, can you see that? So this law of use and disuse is explaining why certain organisms have changed their structure over time. All right. And then Lamarck proposed his second law, which is the law of inheritance, what you gain from your parents, inheritance of acquired characteristic. Now, what is an acquired characteristic? It's a characteristic of physical change which you bring about in your body to suit the environment. You never inherit it. You're not born with it. So what you acquire is what you change. So the giraffe stretched its neck. It was born with a short neck, but it stretched its neck. It was determined to change. It stretched its neck more and more to become longer. Right, so that became the acquired characteristic of the giraffe, the long neck. So what Lamarck said is that the characteristic developed during the life of an individual, that means like the long neck, will be passed on to the offspring. So that acquired characteristic is now passed on to the offspring. So those are his two laws. Now, his laws was not accepted by the scientists. Lamarck's laws were rejected. And why was his theory rejected? Because up till now, there is no evidence that the acquired characteristics can be inherited by the offspring. It doesn't mean that if I have acquired a taste for drinking Coke, automatically my offspring or my baby, when it's born, it will reject milk and only want Coke. So there's no evidence like that, that whatever I change, if I decide now to pull my eyelashes longer and longer and longer, if I pull it, it doesn't mean now that if I pull my eyelashes and becomes long, that my child is going to be born with long eyelashes. So up till now, there is no evidence that an acquired characteristic can be inherited by the offspring. Right. And according to Mendel's law, right, an organism cannot change its DNA. Can you change your DNA? Can you make yourself taller if you're short? No. So an organism cannot change its genotype, which determines its phenotype. So, according to Mendel, it's the genes, the genotype that determines the phenotype, and not the phenotype, what we bring about, what we acquire. It cannot be inherited or passed on to the gametes. Okay, so his laws. Why was Lamarck's theory of law of use and disuse, inheritance of acquired characteristic, rejected? Because it is the genotype that determines the phenotype, and not the phenotype that determines the genotype. And there is no evidence that what we acquire during our lifetime as a characteristic, what we change, will be passed on to the offspring. Right. So now, children, without wasting time, we want to know, how is this applied in an exam paper? How will this theory be questioned? And let us look at this very recent question, right? It came in your June 2024 paper by the DBE, that the stickleback fish are small fish found in ocean. And you must learn to read, learners. You must read. Okay, don't rush. Stickleback fish are small fish found in the ocean and freshwater lakes. So remember, that's two aquatic ecosystem. Oceans mean they have salt. Freshwater means there's no salt in the water, right? So then that lake. So the ones in the ocean have spiky fins for protection against predators. So these fish, picture what you are reading now. It has spiky fins to protect themselves against the predators in the ocean. Until 10,000 years ago, most stickleback fish lived in the ocean. So most of the stickleback fish lived in the ocean 10,000 years ago. But when the last ice age entered, the melting ice created new lakes and some populations swam into the lake. So what happens when the ice melted? A lot of water came on there and freshwater lakes. And what happened is that because there's more water now, the little stickleback fish with spiky fins actually swam into the lakes. Okay? The connection between the ocean and the lakes eventually dried up. So they swam into the lakes. Now they can't go back into the ocean because a part of it dried up. Between the ocean and the lakes now, there was a barrier, there was a land. Okay, so now those fish that stayed in the lakes can't go back into the ocean. So the connection between the ocean and lakes dried up, trapping the stickleback population in some lakes for thousands of years. Over time, the stickleback fish population in the lake lost the spiky fins. Remember, in a freshwater lake, there's not much predators compared to an ocean. In the ocean, there's sharks, there's whales, and things like that. But now in the freshwater lake, there isn't predators. So when those little spiky fish went into the lakes over thousands of years, when the scientists looked at them, they saw this stickleback fish, they no more have any spiky fins. They lost it, it disappeared. So how would Lamarck have explained the absence of spiky fins on stickleback fish in the lakes? How would Lamarck explain it? Remember the law of use and disuse? Right. So here, and the law of inheritance of acquired characteristic. So let's see how we'd say it. State Lamarck's two laws: law of use and disuse, law of inheritance of acquired characteristic. You must swap that off. Now, how would he explain the absence of the spiky fin in the stickleback fish? He'll say, all stickleback fish had spikes. So originally, the ancestral, all stickleback fish, or you can say the ancestral stickleback fish in the oceans had spiky fins. Originally, right? But you can even say ancestral stickleback fish had spiky fins at the beginning. In the lakes, there were no predators. Now, what is there? Now, there's no competition. So that is the selection pressure. In the lakes, there's no predators. So the fish did not use their spiky fins. See, law of disuse. So the stickleback fish did not use their spiky fins to protect themselves as there's no predators. So since they use their fins less and less, the spiky fins had disappeared, you see? And the spiky fins disappeared because they used less and less. There's no predators. So this became an acquired characteristic. The acquired characteristic of no spiky fins was then passed on to the offspring, and the next generation of fish, of stickleback fish, had no spiky fins because he said the acquired characteristic of no fins now, the acquired character of no fins was now passed on to the offspring. It was inherited by the offspring. That's according to Lamarck, and that's how he would have explained it. Okay. Now let's look at another scenario relating to Charles Darwin. Remember, Charles Darwin has a theory of evolution called natural selection. That's how he proposed how organisms are different today. So let us read. You must always read because the answers are also in the passage. In 1986, there was a huge radioactive disaster in Chernobyl, and all humans immediately left the area because of the deadly high radiation. So picture what they're saying. There's a place called Chernobyl, and there was an explosion where there was a lot of nuclear radiation. Okay. Animals exposed to this high radiation developed cancer and died, or they died. So the animals that were there, so humans could live, but some animals that were there developed cancer and some of them died. Okay, almost 40 years later, scientists have been studying the wolves that live in this highly radioactive environment where they are exposed to high radiation. Okay, so remember, there was a radioactive disaster, the humans left, and some wolves remained behind. And then after 40 years, some wolves survived that radioactive radiation. Okay, I mean, some died and had cancer, but some survived. So they found that these wolves, how can this wolf survive in such a highly radioactive environment? How did they survive when others died? So they found that those that survived, the wolves have a mutation that makes them immune to cancer. So how did they survive? They developed a mutation that makes them immune or resistant to the radiation or resistant to cancer. They can fight it off. So they developed a mutation that makes them immune or resistant to cancer. That's very important. All right. Now, use Darwin's theory of natural selection to explain the development of immunity to cancer in these wolves. So what is their favorable trait here? These wolves survived. Why? They had immunity. They had what? They had immunity to radiation and cancer. So the others did not. So let us see how you write it. There was variation amongst the population of the wolves. Now you must explain on the next line what is the variation. Some wolves had the mutation which made them immune to cancer. You see, you can get it from the passage. Some wolves had the mutation which made them immune to cancer, and some did not, as long as you can explain it like that. Or you can say, and some did not have the mutation that makes them immune to cancer. So now, when they were exposed to radiation, right, that's a change in the environment, The wolves without the mutation to cancer or radiation, the wolves without the mutation died. And those with the mutation or immunity to cancer and radiation survived. So those that survived were able to reproduce. Those with the mutation survived and reproduced. And you must say the allele, that allele, not just the characteristic. How are you going to get the character? Via an allele. Passing the allele, so when they reproduce, those that survive, they pass the allele for the mutation against cancer. They pass the allele for mutation against cancer to their offspring. So now the offspring, that means the next generation of wolves there was a higher proportion of wolves which had the mutation of immunity to cancer. The next generation had a higher proportion of wolves with the mutation. Why? Because when they reproduced, the allele for the mutation against cancer was passed on to the offspring, and the next generation of wolves had a higher percentage of them had the mutation against cancer. And that is how they survived. Okay. So here is punctuated equilibrium. Here's a little diagram. And this diagram involves two things. Gradualism, where they say that over millions of years, what is punctuated equilibrium? It explains the speed at which, not why are organisms different, but punctuated equilibrium explains the speed at which changes occur. How fast does it occur? How fast does evolution occur in an environment? That's punctuated equilibrium. And they said it can occur in two ways, the changes. One, small changes or little or no changes. See, look at this horse. It gradually changed from small or short to become big. Small changes over millions of years. Or sometimes it can be followed by long periods of no change, followed by sudden rapid changes. So you can have either little or no change over a long period of time, that's called gradualism, followed by long periods of change, long periods where there's no change, and then suddenly a short period of rapid change. See, long periods of no change remain the same, and suddenly, within just a few years, the short horse suddenly became big and tall. Now that is called punctuated equilibrium, where there are long periods of no change followed by rapid changes over a short period of time. But what caused the change? Due to natural selection. If you don't include that words, due to natural selection, it's going to be marked wrong. So what is this now? Punctuated equilibrium means the speed at which evolution takes place, and it either can happen in two ways. There can be little or no changes for a long period of time, or there could be long period of time with no change, followed by a short period of time with rapid changes due to natural selection. And who proposed the speed at which evolution took place? Who wanted to explain it? It was Niles, Eldridge, and Stone Gould. We say Eldridge and Gould, okay? They proposed the time to which evolution takes place. It explains the speed at which evolution takes place, that it involves long periods where species undergo little change or where populations undergo little change, right? That's known as equilibrium or gradualism. And this can alternate, whereby there could be long periods of no change followed by a rapid change over a short period of time. And that is due to what? Natural selection. Right. Now you need to know what is the difference between natural selection and artificial selection. And this is Bansela marks, children. It's free marks. It's very easy. If they say tabulate, you know that you must put in like a T. Okay, like a T like that. And you tabulate. What do I need to tabulate? Natural selection and artificial selection. And then you put your thoughts in of what's natural selection, and let those words, those writings that you put under natural selection, those facts guide you to what must come on your right-hand side. So let's see, natural selection. You're going to ask yourself, who selects for natural selection? The environment or nature selects. So who selects the trait? It is the environment or nature selects the trait. Then, what is artificial selection? Where humans select the trait. Now let's look at this mealies on the side. Normally mealies had little kernels, like a long mealy with small kernels, okay? Or you could have a big mealy with many kernels. So the farmers decided, I don't want to have a small mealy with big kernels. I want to have a long mealy with big kernels. So they were able to take the genes from the mealy, which is long, and the genes from a mealy which has many cobs and big cobs, and then they were able to pollinate them themselves. The humans pollinated it, not the bees. So humans decided that we want to breed these two different species together. And then when they did that, now they got a mealy which is long with lots of kernels and many kernels. Can you see? So this is artificially selected by humans for their own needs. So who selects in natural selection? Nature selects. Who selects in artificial selection? Humans select. All right. Next one. Now, what is selected in nature? The trait that is most suited to the environment, the trait that can benefit the organism. So, the trait that is suitable to the environment for the organism to survive, that is selected. And in artificial selection, what is selected? Humans select the trait that can satisfy human needs because people want mealies. It's a staple food. They want more and more mealies. So they need to produce a lot at one time. So according to human needs, they need big mealies, big corns, big kernels. So therefore, the selection is according to satisfying human needs. Natural selection, what is selected? Nature selected, human selected. What is selected? The trait that enables the organism to survive in the environment. And then artificial selection, the trait that is selected is the one that helps the humans to satisfy their needs, right? And then where does natural selection occur? Natural selection can occur within one species. It can occur within the same species, only the giraffe. Can you see that? But artificial selection is when you take two different species now and you want to mate them. Okay, you want to take now two different things. You want to take a donkey and a horse, and now you want to mate them. Or you want to take two different species of cows and you want to mate them because you want lots of beef and lots of milk. So two different species. So artificial selection occurs between two or more different species, whereas natural selection occurs within the same species. Okay. And then there's a guideline now to tell you what's the next part of evolution. Can you see it's not difficult? So you need to know what is speciation. How is a new species brought about? How come this tiger has like black stripes and a white coat, and this tiger has a brown coat with black stripes? But we know it's a tiger, not a lizard. But they look different. These tigers are different. We have a white lion and we have a brown lion. How come these species look different? Okay, even with the humans, we are all humans, but each one of us look different. So how come we are looking different? What brings about the speciation? What makes, for example, a white rose different from a red rose? They're different species. So how is speciation brought about? And one of the ways is called geographical isolation. You must know, children, that speciation always increases the biodiversity because we're getting more and more species from an existing one, and that extinction decreases biodiversity due to human impact. That with fires and poaching, we will decrease our animals and plants, and eventually they become extinct. So extinction decreases biodiversity, whereas speciation, you're going to get more species from an original one, and that increases biodiversity. This is a general explanation of how new species are brought about because they were separated by a geographical barrier on earth, like a river, a mountain, a lake, a bed of rock. Okay, all that. So the species became the population was separated to become new species. Now, this is, they just say, describe geographical isolation for 7 marks. Then you just explain it like this: that if there's a single, if a population of a single species becomes separated, so there was a population, right, of this one species that lived on a land, okay? Then the population was split into two by a geographical barrier called a river, mountain, or a lake. Then each population on either side of the barrier was exposed to different environmental conditions, maybe predation, temperature, right, food, competition. So that's the selection. They were separated, and then each one was exposed to different selection pressures or environmental conditions. Then obviously what happens is that in each of these populations now, each of the populations that were separated, natural selection occurred in each one independently. You do not explain natural selection as part of geographical isolation. You just mention that when the one species that lived on land was split into two, then the population of that species underwent natural selection independently because they were exposed to different environmental conditions. And because of that, they became genotypically and phenotypically different, right? They could no longer interbreed because there was a barrier between them. They could no longer interbreed, and each population now became a new species, okay? So that is how you explain. That's a general one. Then we're also going to talk about, for example, that baobab trees in Africa, the proteas in South Africa, the tortoises, the finches in the Galapagos Island. Okay, even the proteas, they look different. The protea in South Africa will look different from the protea in for example, in Madagascar or Australia within the same latitude. And you find that geographical isolation is an example of evidence of biogeography, whereby species or populations were split, okay, due to the river or a mountain, and then they were split. They look the same, but yet they are different. They are new species. All right. And here's an example that was tested from one of the DBE papers, right? The present day distribution. The minute they talk about the study of the past and the present distribution of organisms in various continents, the study of the present and the past distribution of organisms in different continents. Now, you know, continents, geography, living things is bio. So that is an evidence of biogeography, okay? But what brought about the new species is geographical isolation. So they're saying that the present day distribution of three closely related species of the dog family. So here's the dog family. That's the ancestral dog. And then from this dog family now, you find that somewhere in maybe North America, that dog became a coyote. Then somewhere in Africa, it's now known as a jackal. Then somewhere there in Madagascar, it's now called a dingo. But you can see that this doesn't look like a cat. It looks like it's a dog. But it's a dog, but there's something different about it. It means they had coming from the same common ancestor. Now they want to know, how did we get three different species of this dog family from one ancestral species? So what you're going to say? What type of evidence for evolution is represented here? Just remember, children, the minute you see the map and that there's something that looks the same, but they're a little different, whether it's a protea, whether it's an ostrich, whether it's a dog, you must know that evidence is called what? Biogeography. Describe how these three species could have evolved from a common ancestor. So the common ancestor was a dog, okay? Now we're going to say, look at the mark allocation 7. You must learn how to apply that knowledge to that. But they're saying they want to know about the three species coming from the common ancestor. Now that's going to be compulsory marks. So you have to say the original population, you can say the original population of dogs or the common ancestor of dogs lived on a large continent. Okay? The common ancestor of the dog lived on a large continent. Then they became separate. Now, these dogs, they became separated by continental drifts or by oceans. Okay? So you remember there's volcanic activity occurring under the earth, and it causes tectonic plates or continents to move apart. So I'm going to say the ancestral dog lived on one continent. They became separated due to continental drift, or they became separated due to by oceans, right? So that becomes your geographical barrier. So once they become separated by the ocean, now they can't just interbreed because they're living far. Can we interbreed with someone we are living along the Indian Ocean? Can you go mate with someone now, right now, along the Pacific Ocean? No, You are too far. Okay, so there's no gene flow among the three populations. So remember, there was an ancestral dog that lived on one continent. They were separated by the oceans. Due to this, there was no interbreeding or no gene flow amongst the three populations, because they're saying three species. Right? That means the three populations will become three species. So there was no gene flow among the three populations of dogs. Each population now experienced different environmental conditions. Why? They underwent natural selection independently or on their own. Then they became genotypically and phenotypically different. It also changed the genes, and because the genes changed, it changed the outside. They became different genotypically and phenotypically. And if the three populations of dogs were to mix again, they could no longer interbreed because each dog population became a coyote, a jackal, and a dingo. And you can see they had given you another compulsory mark. So first of all, you had to say, because the three populations were separated by the oceans, there was no gene flow, no interbreeding. That's a compulsory mark. And because these three populations, if they were to meet again, they were so different, they became a new species called the coyote, the jackal, and the dingo. See, even if they were to mix again, they'll not be able to interbreed or produce a fertile offspring. They formed three different species, the coyote, the jackal, and the dingo. You had to mention that. So please, children, answer according to what the question needs, okay? Right, and that is practice. Now, there's something else that also separates, that also causes reproductive isolating mechanism, that causes new species. Like, how come a flower like a rose can only be pollinated by the pollen of a rose and not by the pollen of a pine tree, for example? So let us look at these. You need to know it and apply it to the question that they give you in the paper. There are 5 reproductive isolating mechanisms. One is called adaptation to different pollinating agents, right? So if they show you in the picture, in the paper, that there's a flower and there's a bee, then you must know that that flower is only pollinated by the bee. That's why the bee will only pollinate the rose, because it's attracted to the nectar. Breeding at different times of the year, if they give you a calendar like this, and you can see that this butterfly, species B, is more active in, let's say, in September, whereas this one is more active, you can say, in April. So these butterflies, they look the same, but they're different species. They can't mate, because if they do mate, the offspring will be viable, means it can survive, but it's not going to have any gametes. It's going to be sterile. It's going to be sterile. It's going to be infertile. That offspring is going to be infertile. So 2 different species cannot mate because the offspring is going to be infertile. Only when they belong to the same species and if they mate, then the offspring will be fertile. So how do you know whether 2 individuals belong to different species? If those individuals mate and the offspring is fertile, then they belong to the same species. If they mate and the offspring is infertile, you know they are different species. And what could cause it was that, or what can make them separate? We don't want to have individuals which are infertile. So how does nature ensure that you don't have individuals that are infertile, otherwise become extinct? Okay? So how does nature do it? It ensures that if you're a different species and then you're a different species, you can't mate at the same time. So you will mate in April, you will mate in September. So breeding at different times of the year. Okay? So that when they do mate, they mate with the same species. Then you have here also a courtship behavior, behavioral patterns, okay? For example, a duck will quack and quack and quack, and that's how it will attract another male duck. But you will find the peacock will open its feathers to attract the female. So you have different courtship behavior. So obviously the peacock cannot mate with the duck because the peacock will not understand the behavior of the duck quacking. But only another peacock will understand the peacock's behavior, that when it opens the feathers, it's time for mating. And that's why peacocks will mate with peacocks, okay? Infertile offspring, that if 2 different species do mate, then the offspring is going to be infertile. So that's another reason why two different species will not mate, all right? And you can see here a donkey and a horse, they will actually form a mule, and a mule is infertile. Okay. So also the fifth one that keeps species separate is the prevention of fertilization. And you can see this is a very small dog and this is a very big Dane. And the reproductive organs of that small little dog cannot mate, or it's not compatible with the reproductive organs of this big dog. So this is called prevention of fertilization. Sometimes distances can also be a method of prevention of fertilization. So you must learn the three reproductive isolating mechanisms that keep species separate. And you must know, what is a species? It is a group of organisms that are able to interbreed, and they have the same characteristic, and they produce a fertile offspring. Whereas the population is a group of organisms that live in one area at the same time, at the same place, capable of interbreeding, and that's it. Okay. So let's go to human evolution. Now, this is another part of evolution, and it can be very simple, children. Here we have the humans, and here we have the African apes or the gorillas. And what are the similarities? They can ask you to list 3, they can ask you to list 4. So let us start off let us see what is similar to both of them. Both the humans and the African apes, that means your Australopithecus, your Ardipithecus, okay, our primates, they had an upright posture. Okay, our primates, between humans and our primates, that is the human ancestors, they had an upright posture. Also, they had a large brain relative to the body mass. So our brain compared to our body mass was quite large compared to any other part of the body. The brain is quite heavy and big. So we also have a long upper arm. So Australopithecus, Ardipithecus, they have a long upper arm, they have a large brain, they have eyes in front, right? Eyes with cones to see color, they could see color. The eyes face forward, they could have binocular vision where they can focus at an object in 3D. Also, humans and our primates had a poor sense of smell, because we are not animals like a dog or a cat where they have to rely on instinct to look for food. They have a very good sense of smell. But humans and our primates have a poor sense of smell, or they say olfactory brain centers are reduced. Humans and our primates have 2 mammary glands, okay, unlike the dog, which may have 5 mammary glands. We have freely rotating arms. Okay, our rotation around the elbow joint and wrist is the same. The rotation around the elbow joint and wrist joints, okay? It's 180 degrees. And then we have bare fingertips. And then we can say we have nails instead of claws. It's the same thing. You don't get separate marks for that. It's one and the same. Bare fingertips, or you can say we have nails instead of claws. And we also have opposable thumbs. So those, you can learn them, and those are the characteristics that have similar to humans and our African apes. And then the differences between humans and African apes. What's different between now humans and chimpanzees or gorilla? Now here, we as humans, we walk on 2 feet. So we are bipedal. Whereas the gorilla and the chimpanzee walk on their hands and feet. So they're quadrupedal. That's the method or mode of locomotion. So how come we are able to walk on 2 feet? Looking at our vertebral column, that means the vertebral bones with the spinal cord, it's upright, it's S-shaped. So we have an S-shaped spine that keeps us upright. Okay? Our foramen magnum, the opening where the vertebral column enters, think of it like a lollipop, a stock sweet, where the sweet itself is the brain. And from the bottom there, where the stick enters, that opening, it's called the foramen magnum. So our foramen magnum now moved from the back, and it moved forward. It moved to the bottom of the skull, so that the vertebral column can enter from the bottom. So foramen magnum moved so here's your skull. Our foramen magnum moved from the back, from the back here, it moved in a forward position, okay? So that our vertebral column can enter from the bottom. Whereas the African ape, you can see that the foramen magnum is at the back, okay, like a dog, like that. For the gorilla, it enters from the back, okay? Whereas ours, it enters from the bottom, because our foramen magnum moved in a forward position. So let's look at the cranium. The cranium is the bone that protects the brain. In humans, we have a large brain, so humans will have a large cranium. Gorillas will have a small cranium. You can't say larger and small. You must either say what. If you have a large brain, then the chimpanzees have a small brain. Or if you say humans have a larger brain, then chimpanzees have a smaller brain. Okay? The palate shape, the shape of the jaw, ours is semicircular like that for humans. And you find the gorillas have a rectangular palate shape. Okay, the shape of the jaw or palate is rectangular. Then the dentition, we as humans don't have gaps between the teeth called diastema, whereas the gorilla has gaps between the teeth. Okay, that's called diastema. We have small little canines, we have small canines, whereas the chimpanzee has large protruding canines, right? Long pointed canines. See, larger canines. Humans, here by our chin and our jaw, we have a flatter jaw. So a flatter face and non-prognathous. Whereas chimpanzees have a protruding, the jaw is what makes it prognathous. So they have a prognathous jaw. Okay, prognathous jaw means the face slopes because of the jaw, the large jaw, large teeth. It makes it prognathous. And humans have a flatter jaw, which means non-prognathous. And then the humans, we don't have a ridge on our cranium and we don't have a protruded brow ridge. So our cranial and brow ridge is absent or less protruded. Whereas in the gorillas or African apes, the cranium has a ridge and the brow ridge is quite prominent or protruded. Okay, you'll see this in your SIP notes. Please study the differences on the diagrams of the skull, the pelvic girdle, that means your hips, short, wide hips, long, narrow hips, and pelvis. Okay, you need to know that. And here is the diagram. This is the whole skull of a chimpanzee. Immediately you can see the rectangular jaw. You can see the large pointed canines. You can see this opening here is the foramen magnum, and you can see it at the back. Okay, here you can see this is a C-shaped palate or jaw. You can see no gaps between the teeth. Here you can see the gaps between the teeth. Here's the gap between the teeth. But here we don't have no gaps between the teeth in the human. It's a C-shaped jaw, right? Gently curved jaw, C-shaped, rectangular. Large pointed canines, small canines. Foramen magnum at the back, foramen magnum. Can you see it moved forward here? Here it moved at the back. That's quadrupedal, that's bipedal. Look at the hips now, or the pelvis. C. This human has a, so C will actually go with the human skull. And this gorilla goes with that pelvis. So here, this is the long, narrow pelvis of the gorilla because it also puts its body weight on its pelvis, right? So let's see. Tabulate 3 observable, what you can see. Differences between skull A and B. What can we see there? Let's see. We can see skull A and B. Now, you must already label it on your question paper. Gorilla or African ape? Humans. Humans, African ape, you know that. So let's look at you, and you always compare the same things. Rectangular jaw, C-shaped jaw, or semicircular jaw, right? Then, or you can say rectangular palate, or C-shaped palate, or semicircular palate. Let's look at the canines. Long pointed canines, or large canines, small canines. Then let's look at the foramen magnum. Foramen magnum is at the back of the skull, and then the foramen magnum moved in a forward position. Okay, you can see here the jaw is large, small jaw. So that's the size of the jaw. The other one was about the shape of the jaw. Okay, what else do you see here? This, you can't talk about short, wide pelvis and narrow pelvis here because they are talking about the skull. So don't make a mistake and start comparing the pelvis in this table because we want to know about the skull. And you can see here that here the jaw is more, can you see it's more protrude? So prognathous jaw, less prognathous jaw. All right. State why pelvis C belongs to a human, because the minute you do this, you must look for something that looks like this. Short and wide. This one here is long and narrow. So D is a gorilla or African ape, and that one's a human. So state why C belongs to a human is short and wide, okay? For efficient locomotion, because most of the body weight is on the feet. So it's short and wide to offer more strength for efficient locomotion. Now, we as humans and our primates, We belong to the family. That's the most important. Hominidae. So humans, together with the gorillas and the African apes and the Ardipithecus and also humans and the hominids and the hominins, right? Hominins are human ancestors. Hominids are the ones with the tail, the chimpanzee and the gorilla. So humans and our primates, we all belong to what? The family Hominidae. It always comes in the section A. Right, let's look at a phylogenetic tree, children. The phylogenetic tree is like a tree. What is it? It looks like a branch like this. Am I right? Ne? A main branch and then small side branches. So what is a phylogenetic tree? It's a schematic form, right? It's like a flow diagram that shows the relationships within a set of organisms. It shows you the relationship between closely related and not so closely related organisms, right? And what else does it show you? It shows you when will an organism become extinct and when does an organism originate. So it's a group of organisms and the relationship between them, and it must not be confused with the pedigree diagram in genetics. So remember, a phylogenetic tree tells you who's the common ancestor, who is extinct, when did they become extinct, when did the species originate, and how long did it live for. So you got to see when it died out. You got to have a ruler and a pencil for this. See when it originated. Obviously, something originated 5 million years ago. It can die of 2 million years. You can't originate 2 million years ago, but you died 5 million years ago. So just remember the phylogenetic tree works like that. Okay, so if you have to have a tree like this now, you can see if this is 6, 5, 4, 3, 2, 1, and an organism is here. So that means that's where it originated about 5.5 million years ago, and it died of 5 million years ago. So 5.5 minus 5, it means it lived for only 0.5 million years on Earth. So the starting line is way before, that's when it originated, and where it stops for that species, that's when it becomes extinct, and you subtract the difference to see how long it lived on Earth. So here's an example from a paper that just was recently tested. Give the letter representing the common ancestor for all the species shown in the phylogenetic tree. So let's see, all these are different species. Who's the common ancestor? It's going to be V. V gave rise to all of them. So the letter is going to be V. Can you see that? Next one, identify the species that's most closely related to old world monkeys. So look, where's old world monkeys? There's it. And then who's most closely related to it? Let's see there. Right. It seems like the one that's closely related to it is going to be the gibbons. Can you see? Because old world monkeys comes there, it stops there, then from there it's that one. So it seems like gibbons and old world monkeys are more closely related because they share the common ancestor called U. Can you see? So it's going to be the gibbons, right? That's the species that's most closely related. It's going to be the gibbons because you see old world monkeys, that line stops there and it branches off to that. So this one gave rise to gibbons and old world monkeys. And the common ancestor there was U. So state 3 characteristics of the upper arms or upper limbs that humans share with group P. So who's group P? Chimpanzees, gorilla, and orangutans. What do we share with our primates? We have long upper arms. We have opposable thumbs. They also have they can also hold a banana. So they can also hold a cup. So we have opposable thumbs. We have long upper arms. We have freely rotating arms. All right. And all those that we have mentioned earlier on, those things that are common to us, we have 5 digits, 1, 2, 3, 4. We have 5 digits, so 5 fingers, 5 digits, pentadactyl hand, we have opposable thumbs, we have nails instead of claws, we have bare fingertips. Our rotation of the wrist is now 180 degrees, our rotation of the elbow is 180 degrees, and we have longer upper arms, and we have freely rotating arms, and we have eyes in front. So those are the things that we as humans share with the chimpanzees, gorillas, and orangutans. Identify 3 species that share the common ancestor R. There's R now. Now who is also sharing, who's the 3 species that share R as the common ancestor? It's going to be the gorillas, right? From R comes what? The chimpanzees, right? And then the humans. Can you see that? R. He's a common ancestor for gorillas, for Q, for chimpanzees, and for humans. So it's going to be chimpanzees, humans, gorillas. And so we're going to look at R and we're going to see it goes there, then from there to there. So all that for Q, humans, chimpanzees, and gorillas. They are the three species that have R as the most recent common ancestor. Actually, Q is the most recent common ancestor for humans and chimpanzees, but R is a common ancestor for that whole group there. Okay, right? Because it's a common ancestor, and that's going to be R for 1, 2, 3. Now let's look at this one here. It's the 3 lines of evidence, right? Used for a common ancestor. How do we know that we have a common ancestor? The 3 evidence is fossil evidence, and you can see the Great Rift Valley in East Africa, in South Africa, the cradle of humankind. We have fossils that indicate that we are humans and we have human ancestors. There's fossil evidence to indicate that trend. Genetic evidence is that if we take the mitochondrial DNA mutation of present-day females, maternal, female ancestry, because mothers give birth, men don't give birth. So mothers give birth to children, boys and girls, but you've got to also look at the girl, the female part, right? So if you look at the mitochondrial DNA in the cells of all females of any race throughout the world, the mitochondrial DNA mutation of present-day female matches the mitochondrial DNA of the oldest female fossil, right? So there is genetic evidence that if our mitochondrial DNA matches the oldest female fossil that came from Africa, it means we have a common ancestor. That's why our DNA in the mitochondria is the same. Cultural evidence. We are using knives. We have knives. We have spears. Those tools that we make today, it's coming from the tools that were made by Homo habilis many years ago. So cultural evidence, genetic evidence, and fossil evidence is there to say that the tools that are made now are modified from what the tools were made millions of years ago. Okay, so they used to use stone tools, right? And the mitochondrial DNA, remember DNA has genes, so that is genetic evidence. And we can see fossils in the cradle of humankind. Now, this is the phylogenetic tree, and you can see millions of years this is present. So Homo sapien is human. This is Homo neanderthalensis, Homo erectus, Homo habilis, Homo Australopithecus africanus, okay? So how many genera are shown in the diagram? Genera means plural of genus. So we can see Australopithecus africanus. Australopithecus, A stands for Australopithecus, that's a genus. That's Homo, 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 Homo. So although the species in small letters are different, but it's the same genus. So how many genera are shown here? That's 1, 2, 3, 4. 4 homos, which means it's the same genus. That's 1. And this is Australopithecus, that's the second genus. So how many genera are shown here is 2. But how many different species are shown? 1, 2, 3, 4, 5. Different species. How many different species of Homo are shown? 1, 2, 3, 4. How many species of Australopithecus is shown? Only 1. But how many genus is shown? That's a genus Australopithecus and that's a genus Homo. So altogether, it's Homo genus and the genus is Homo and the other genus is Australopithecus. So there's 2 genera. Genera means the plural of genus is genera, more than 2. And we can see 2. When did Australopithecus africanus become extinct? So you take your ruler, you say, oh, he originated around. Always calibrate and say that is like 3 to 3.5. So that's about 3.250. So that looks like 3.1. So he originated 3.1 million years ago, and then he just stopped here at 2. So he became extinct how many million years ago? 2 million years ago. So it's very easy to analyze a phylogenetic tree. And the same tree again, explain the influence of brain volume on the complexity of tools made by Homo habilis and Homo sapiens. Don't get scared for a question like this, children. It's nothing different. They're only asking you that the brain volume, right? The brain volume, look at the brain volume. They're telling you Homo sapiens have a brain volume of 1,350 cubic centimeters, right? Homo neanderthalis has a brain volume of 1,500 cubic centimeters. So now the brain volume of the Homo species is quite big compared to Australopithecus and Homo habilis and Homo erectus. So over time, the tools became more complex because the brain volume increased. So as the brain volume increased, it means more intelligence and therefore more tools became complex. So Homo sapiens have a bigger brain volume than Homo habilis. Can you see that? Homo habilis was called the handyman, the tool man. So Homo sapiens' brain was bigger than that of Homo habilis, right? Therefore, Homo sapiens were more intelligent or had a greater brain capacity and made them more intelligent, leading to Homo sapiens developing more complex tools and specialized tools compared to Homo habilis who only made stone tools. Right, next one. Here is the question. It shows you that the evidence of fossils, Homo erectus, shows that they appeared in Africa 2 million years ago. So Homo erectus fossils appeared in Africa 2 million years ago. Then they spread, okay? They went out they were walking over the ice. And Homo erectus is the oldest known species to be fully bipedal and have a human-like body. So he's bipedal, he has a short white pelvis. Their brain size was smaller than that of humans, and a prominent brow ridge was present over their eye sockets. Okay, so let's see. Describe how all other fossil evidence is used to support the out of Africa hypothesis. What are we going to say? That the oldest Homo erectus was found in Africa, and younger ones were found in other continents. Okay. Also, and the oldest Ardipithecus fossil was found in Africa. The oldest and the only Australopithecus was found in Africa. The oldest and the only Homo habilis was found in Africa. The oldest Homo erectus was found in Africa, but the younger ones were found in other continents. That's Homo erectus. Even Homo sapiens. The oldest Homo sapien was found in Africa, and younger Homo sapiens were found in other continents or other parts of the world. So that is fossil evidence. Then describe how fossils of Homo erectus was used. As I said, the oldest Homo erectus was found in Africa, whereas the younger ones were found in other parts of the world, indicating that Homo erectus, if he was the oldest here, he moved from here. He moved out of Africa to other continents. And then use the information regarding Homo erectus and tabulate two differences between the features of Homo erectus and Homo sapiens, and you'll get it. Because they're saying you must use the information in the passage, so you must read. And you must tabulate, because they're saying tabulate. So they want what? Homo erectus and Homo sapiens. So let's read that Homo erectus, the brain was small, so small brain compared to humans, they say, than that of humans. Then what else do they say? That Homo erectus had a prominent brow ridge, and Homo sapiens had a reduced brow ridge. Okay, what observation of the skull of Homo erectus would have led scientists to conclude that Homo erectus was bipedal, right? What could they have seen in the skull of Homo erectus, not in the pelvis, in the skull? And what tells you are bipedal in the skull? The foramen magnum moves in a forward position. That tells you you're bipedal in the skull. And the other thing that tells you you're bipedal is the S-shaped spine, if we talk about the spine. And the other part that tells you you're bipedal is the short, wide pelvis, okay? So read the question properly. Which part do they need you to refer to? Okay, and now we're going to genetics, children, right? And that is, I told you, 48 marks. And then let's see, you need to know these words like genotype is you actually using alphabet which represent alleles. Phenotype, you must use words that describe the appearance. Okay, now you must learn the recipe or format of a genetic cross. And if you don't, you're going to get it wrong in the exam. So you must put P1 on your left-hand side where the marker can see it. P1, first parents. Then you say P1 and then phenotype P means describe what you need to put there. Is it a round worm and a flat worm? Is it a tall man and a short woman? Man with blue eyes, man with brown eyes? What is the physical trait? So you write it in words, the phenotype. Below it, there's enough paper. Don't squash your work. Below it, write. Below phenotype, write genotype. Oh yes, I must use words. I must use alphabets. I must use letters. Big letter or small letter. Small letters are recessive. Big letters are dominant. I must put it, if it's homozygous dominant, I must use 2 big letters. If it's homozygous recessive, I must use 2 small letters, because that's what's going to show in my phenotype. And then also, if I'm going to use heterozygous, big and small letters, okay? So you write down the genotype using your alphabets. Then below that, you must write meiosis. And below the meiosis, you must write down the gametes. And you separate them by a comma. You must write, there'll be one alphabet coming from the 2 genotypes. You'll spread them out. The gametes will segregate, okay? And then you'll separate them. Then you do a cross, or you use a Punnett square. A Punnett square is bigger. You just make 3 rows and 3 columns. And here you're going to write gametes, and you're going to put 1 gamete per block, like big G, or small g, small g. So you know that's your gametes. You're putting your gametes there, and you're putting your gametes there. And here are going to be a 4 possible offsprings. It doesn't mean the parents are going to have 4 children. It means these are the 4 possible genotypes of the offsprings. The alleles, the genotypes of the offsprings will be there. So whether you have 20 children or 1 child, those children have any of that 4 possibilities of genotypes. All right. So after you draw the Punnett square, you must write down what are the genotypes of those offsprings. You'll put those offsprings are F1. What is the genotypes? Now, looking at the genotypes, describe the genotypes, and then find the ratios or percentage. Remember, you're going to get a mark for P1 and F1. You're going to get a mark for meiosis and fertilization. That's already 2 out of 150. You are looking for 43 marks to pass that paper. So that's very simple, children. And then what you see, phenotype, I said it's words. Genotype, you're going to say it's the alleles or your alphabet. And the alleles are your alphabet. And you'll see they'll separate here to form your gamete, which is represented by 1. So if a genotype is TT, then your gamete is going to be big T and comma big T. Can you see that? If your genetic makeup is small t, then it's going to be small t, comma small t. All right. So during fertilization, the individual gets 1 allele from each gamete. You do not say that the individual gets 1 B and 1 T. Are we getting it from the dictionary? No, you're going to say it gets 1 allele for tall and 1 allele for short. You say allele, you don't say it gets 1 X and 1 Y or 1 T and 1 small t. You must say it's the allele, okay? Now we're going to look at monohybrids, which means it's the cross that talks about 1 genetic trait. Complete dominance, codominance, and incomplete dominance. Whereas when we talk about dominance, we don't talk about small letters here. It's either big R, like red flower and red flower crossed over with white flower, white flower. Codominance is where the pink flower, the red flower, and the yellow, the red flower, and let's say the yellow apple gives you a red yellow apple. Both the alleles are together present in the phenotype. Incomplete dominance, you're going to get an intermediate phenotype, whereby a red flower is crossed with a white flower, and now the flower is RW, it's pink. It's not red, it's not white, it's completely different. It's called incomplete dominant because there's an intermediate phenotype, and we use only capital letters for that cross. So here's an example of codominance, children, right? And a farmer has an orchard of apple trees, and each apple's produced there was red yellow. To extend his apple orchard, the farmer collected seeds from the red yellow apples and grew them. When the new trees matured, he found that some of the trees produced red apples, others produced yellow, and the rest produced red yellow apples. So use the genetic cross to explain his results in the F1. So how did the farmer who took red apples and yellow apples, when he crossed them in the F1, they were red apples, red yellow, and red apples, red yellow, and yellow apples. Now, so now, how are we going to explain that genetic cross? We're going to say P1 phenotype red yellow, red yellow. There's red, yellow. Okay, can you see? And then you're going to have now, you're going to show how he got it. He said red. He took the two red, yellow apples. And he took a red, yellow apple. That's meiosis. That's the gametes, R and Y. You can see you separate them. Then you put them into a cross, and you can see that's how he got his red apples there. He got his red, yellow apples, right? And he got his yellow apples there. And that's how you got it, P1, F1. Okay, now what proportion of apples in the F1 will be red, yellow? You can see, children, this is altogether 100%. So that's 25% red apples, 50% red, yellow, and 25% yellow apples. So 50% will be red, yellow. And here's your red apple crossed with the yellow apple, and it's giving you your red, yellow apples. Then they said here, a farmer only wanted to sell red, yellow apples. He is selling very well. So what must he cross to get only red, yellow apples? He mustn't take red, yellow, and cross them. He must just take a red apple and a yellow apple, and he'll only get red, yellow apples. So the farmer saw that the red, yellow apples are the best. Name the phenotype of the trees that he should cross in future in order to ensure that the new trees will produce only red, yellow apples. A tree with red apples must be crossed with that of yellow apples. Okay, and here's the dihybrid cross. A dihybrid cross. And this is, when you talk about dihybrid cross, you talk about Mendel's law of independent assortment, because the alleles of two different genes separate independently into the gamete, whereby a gamete can get a tall allele and a purple flower, and a gamete can get a short allele and a yellow flower. Can you see that? So when the alleles separate independently for a genetic trait, it's the law of independent assortment. And children, you're going to look at this worksheet. You're going to actually see also this in the question paper, right? We're going to give you this, and this is talking to you about the gametes, right? That here you can see if it's a heterozygous of two characteristics, then the parent must be heterozygous for both traits. It's going to be a big F for short fingers and small f for long fingers. Widow's peak is dominant. That's capital H. So continuous hairline will be small h. That's heterozygous. And that's the genotype of the parent. And the genotype of Z, you can see, you can take that small f and that small f, you put it there and there, and you put the alleles together. Now you don't put F and H, you keep the alleles or the same alphabet together. F and F gives you FF, big H and small h, and that's going to be the genotype of Z. Okay, and you can see here it is. And the number of genotypes that could result in offspring with short fingers and continuous hairlines. So you can see short fingers is a big F and continuous hairline is a small H. So you're going to look for everything that has a big F and a small h. So that is going to be big F and small h is 1, it's going to be 2, and it's going to be 3. Can you see that? So that's 3. Allele for continuous hairline, if widow's peak is big H, allele for continuous hairline is going to be small h. Phenotype of a child who is homozygous recessive for both characteristics. What is going to be a phenotype of a child who is homozygous recessive for both characteristics? It's going to be what? Homozygous recessive. It's going to be long fingers with a continuous hairline. You see? Because that's going to be small letter, small letter. And here, children, it's very simple. We need to know that we have 46 chromosomes, 23 from the mother, 23 chromosomes from the mother, 23 chromosomes from the mother. And out of the 23, 22 pairs are autosomes, and 1 pair is a gonosome in males. The XY is a gonosome in females. The XX is a gonosome on pair 23. And this is the sex determination of the karyotype, where we see the autosomes from pair 1 to pair 22, giving you the shape, size, and chromosomes arranged in pairs. Right, that's autosomes. And the last pair tells you the gender. If it's two big X, it is XX, it's a female. If it's a big X and a small X, what's the small little one? That's an XY, which is a male. Right, and here is how you work out the sex determination. You can see you must always separate the gametes with a comma, and you always keep the genotypes. When you're looking at this, you keep the genotypes of the alleles together like that. You separate them like that. Don't separate them like that. It's going to be wrong, because then that looks like a gamete. So you're looking at a genotype of offspring. You keep them together like that, not separate like that. Okay, you only do that with the gamete where they are separated, and you describe them in the phenotype. All right. This is the sex-linked inheritance, children, that we got a lot of time now. We got to do a lot of work just to know that when there's a mutation on a chromosome of the 23rd pair, you call that a sex-linked disorder or inheritance, like color blindness or hemophilia, whereby on the X chromosome, you say X to the power big D or X to the power whatever they give you there. When it's the X to the power something, you know it's a sex-linked disorder. Okay, you must know if that disorder is going to be dominant or caused by a recessive allele. And you can see hemophilia here, that when you are receiving, when a child inherits it, it's going to be, okay. So hemophilia is a genetic disorder caused by a recessive allele on an X chromosome. Right? The hemophilic female marries a normal male. Explain why all these sons will be hemophilic. So remember, children, hemophilia is where a male, remember that a male is XY and a female is XX, right? So now the Y chromosome doesn't have an allele. So if a male is hemophilic and is caused by a recessive allele, and let's say I want to use now for hemophilia, I want to use small h, then that means he's a hemophilic male. But a female, for her to be hemophilic, she has to have on both her X chromosomes, X to the power small h, X to the power small h. It's caused by a recessive allele on both her X chromosomes. So there's no capital letter or dominant one that's masking this recessive, this one that's causing the disorder. So for the female, on both her X chromosomes, she must have the 2. And also learn, the children, that when you inherit from the parent, the father with the Y, the male, he donates his Y chromosome to the child. And the child must now get an X. The X can only come from the mother. So if it's a boy child, he's a boy, he's a son, because he's getting the Y chromosome from the sperm. And the other X is coming from the ovum. Can you see that? The allele. All right. If it's a girl, it means that the father is donating his X. He cannot donate his Y because then he becomes a son. So when it's a female child, the female child is getting the X. The X. You remember your gender, you are XX or XY at the end to tell you whether you're boy or girl. So how did you get that XX if you're a girl? One chromosome comes from the mother, that's the X, and the other chromosome comes from the father, it can only be the X. Because if you get a Y, then that means you're a boy. So okay, so your individual inherits one allele from each parent. So I can inherit this allele here from the father because I'm a female, and then my other allele can come from the mother. So if the mother, if my father is hemophilic and my mother is hemophilic, I'm going to get the XH from my father because I'm a female, and I'm going to get the other X small h from my mother, and she's hemophilic. So I'm also going to be hemophilic like my parents. But if my mother she's normal, but she's a carrier, she's heterozygous normal. For genotype, you don't say heterozygous, you don't say carrier, because you can't see a carrier in the phenotype. So in the phenotype, you cannot say carrier, okay? And in the phenotype, you cannot say heterozygous. So now, in the question, they will tell you that so that you know how to represent your genotype. If they say that the mother is normal, but she's a carrier of hemophilia, or she's heterozygous normal, and normal is X to the power big H, that means she's heterozygous, she's normal, but she's carrying the hemophilia allele. Now, if I'm going to be a female who's normal, it means I'm getting my one X small h from my father, but I'm normal because my mother's dominant allele for normal is coming to me. Okay? So I can be a normal girl, but my sister can be hemophilic because she may get this X to the power small h from the mother, and obviously that one's coming from the father. So you can be in a family, you have 2 girls in the family, sisters. One sister can be normal, one can be hemophilic. Okay. And remember that the Y chromosome comes from the father, you inherit it, and that the recessive allele is inherited from the mother. Mother has 2 recessive alleles on her 2X chromosomes. A son needs to get only 1 recessive allele. Can you see? A son needs to get only 1 recessive allele to be hemophilic. And he gets that from who? From his mother. A son gets his X chromosome from the mother, and he gets his Y chromosome from the father. Please remember that. The Y chromosome does not carry any allele. Now let's look at color blindness. It's a genetic also caused by a recessive allele on the X chromosome. Children, this is very important. A colorblind man marries. For marriage, you must put a small x. If you don't put it, it's going to be marked wrong. So you put P1, colorblind man, marries a normal vision woman. You cannot put colorblind man cross normal woman. Normal in what? What is normal? Normal vision, because colorblind got to talk about the eyes. So the colorblind male man marries a normal vision woman who does not carry the allele for color blindness. So she's normal, but she's not carrying the allele for color blindness. What does it mean? She is homozygous normal because she's not carrying the allele for color blindness. So do a genetic cross to show the possible phenotypes of the children. Use big B for normal and small b for colorblind. But you know that colorblind is a sex-linked disease. So you can't just put big B, small b, because colorblind is a sex-linked disorder. So how will you represent it? You put P1 phenotype, colorblind man marries a normal vision woman. And what did they say? She does not carry the allele for colorblind. So they are trying to tell you, if it's a colorblind man, he's XY, he's colorblind, so he's going to carry the recessive allele on his X chromosome, and they said you must use small b for colorblind. Then therefore, this mother is not carrying the allele for colorblind. She's completely normal vision. So she's going to be XX because she's a female, and she's going to be absolutely homozygous normal because she's not carrying any allele for colorblind. So she's going to be X to the power big B, X to the power big B. You keep it close. That's the genotype. You're using letters. Then you must write meiosis comes there. And then after meiosis, you're going to put the gametes. You separate. That gamete goes there, comma. That gamete goes there, comma. That gamete goes there, comma. That gamete goes there. You see, you separate the gametes. The gamete, you must see them separate for you to get a full mark. Then what comes next? Fertilization. How will you show fertilization? You'll show it using a Punnett square. 3 rows, 3 columns. What you write here in the top left-hand corner? Gametes. Now put 1 gamete per block from your left. Put that gamete in this block. Put that gamete in that block. Take your gametes on your right. Put 1 gamete at the bottom and 1 gamete at the bottom there. Now you've got your gametes, right? You put them in your Punnett square. Now you fertilize them in any order. That's called random fertilization. That's why fertilization must be in line with your Punnett square. You say, if this gamete fertilizes that, I'm going to get an offspring like that. X, 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 X. You always put the big letter first. X to the power big B, X to the power small b. Because X to the power big B is normal, this female is going to be normal, but she's going to be carrying the colorblind allele. Can you see? So she's heterozygous normal, right? Genotypically. But phenotypically, she's going to be normal. You can't see she's carrying color blindness. Okay. But she is normal. But hidden in her body is in the genes is the colorblind allele. And then here, if in case this one fertilizes that, that's the offspring. If that gamete fertilizes that one, then it's going to be XY. So this is going to be a boy. And this boy is going to be absolutely normal. All right? Can you see that he's going to be normal? Because he inherits the Y from the father and the B from the mother, X to the power big B, which is normal. So he's normal. And then we finish the first with the first and the first with the second. We finish that gamete with those 2. Now, what about this gamete? If this gamete fertilizes that one, it's going to be XX, X to the power big B, X to the power big B. So even this girl is going to be phenotypically normal, right? But she's heterozygous normal, unlike the mother who was homozygous normal. And then this and this one, it's going to be X and Y. And this male is going to be very, very normal, just like the mother, because the Y comes from the father and he's going to inherit the X with the big B, normal vision, from the mother. So genotypically, you must say F1, that who are these? This is my F1. Write them down separately from your square. It's X. Write them close like that, okay? Close. X to the power big B, small b, comma, or semicolon. Then write that one. then write this one, write that one, separate them. You know, that's the genotype. Now describe them. What is this going to be? It's going to be two females and both are going to be normal. Two normal females. Two females with normal vision. And then here, two males with normal vision. So two normal vision females, two normal vision males. And what is the ratio? 2:2 simplified is 1:1. That means 50% females are normal vision, 50% males are normal vision. All right. And you know you're going to get marked for P1, F1, meiosis, fertilization. Okay. So just as blood groups, children, phenotypically you say I'm blood group A, I'm blood group B, I'm blood group AB, I'm blood group O. That's phenotypically, you can describe that. Blood group A, B. But the alleles, the genotype for that is going to be, I can be homozygous blood group A. In other words, I inherited, for blood groups, you must use I to the power A, not X. When it's X, it's a sex-linked disorder. When it's I to the power A or B, that is blood groups. So this is the allele. They say, write down the allele for blood group A. Write down the allele. So you're going to say I to the power A. Write down the allele for blood group O. You're going to put small I. Okay? That is an allele for blood group O. That is an allele for blood group A. If they say, write down the alleles for a homozygous blood group A, then you're going to put this. They'll say, write down the blood group of a heterozygous blood group A. Then you're going to write that. Okay, so this, write down the alleles for a person with blood group A. You're going to put that. But write down the phenotype of a blood group A. You're going to say blood group A. Write down the genotype of blood group, a genotype of a person with blood group O. You're going to put two small letters. That's the genotype of a person with blood group O. That's a genotype. But the allele for blood group O is just that. Okay, so you need to know that, children, that there are 3 alleles that control 4 blood groups. Blood group A, blood group B, blood group AB, blood group O. So 4 blood groups controlled by 3 alleles. That's why blood group is an example of multiple alleles, more than 2, A, B, and O. So which alleles control blood group? A, B, and O are the 3 alleles that control the 4 blood groups. Which blood group is recessive? It's the O blood group. So O can give blood to any person. Blood group O person can give blood to anybody. But blood group A can't give blood to anybody. Okay? Blood group A cannot give blood to O. But O can give blood to anyone because it's recessive, it's unaffected. And AB, there is no O there at all. It's AB, it's codominant. So blood group is an example of codominance, whereby both the A and the B alleles are equally expressed in the blood. So that is complete dominance, where it's A, blood group A dominates and blood group B dominates. But here A and B dominate. That is an example of co-dominance, and this is a recessive blood group. And that's complete dominance, only O. Right. A man with blood group AB and a woman who is heterozygous for blood group B plan to children. So let's see, how would I say blood group AB? It's going to be I to the capital A, I to the capital B. That is the blood group for men. For a man, please write down somewhere in your question paper at the back where try to work it out first before you write it. And he marries a woman who is heterozygous for blood group B. Heterozygous means she's blood group B, but hetero means she's going to be I to the power big B, and she's going to be carrying the recessive allele. So she's going to carry dominant and recessive for her to be heterozygous. And you represent the small letter O for the recessive. O is represented with a small I. How many alleles control blood groups, inheritance of blood groups? 3. That's I, A, and O. Describe the type of dominance that occurs in the inheritance of blood group B. She's blood group B. B is dominating. So that's complete dominance, because the allele for blood group B is dominant over the allele for blood group O. O is a recessive, but she's blood group B, not O, because B is dominant. So this is complete dominance, okay? And how can we show the genetic cross? P1, you see phenotype, blood group A man, marries. This is marriage, you must put that cross. Marries a woman with blood group B. That's the phenotype. Can you see? We just say blood group B. Genotypically, AB is represented as I to the power A. That's married, I to the power B. Heterozygous B. That's meiosis. Then the gametes, you separate. Can you see how you separate the gametes? Then you put fertilization. Then you're going to put your Punnett square to show them. You take one, what you're going to write here? Gametes. You put one gamete per block, one gamete per block. You write, what are these? The offsprings. Who are those? That is the F1. Can you see? That is the F1. You rather put it there. So you get your mark. That's the F1. Describe them. Put F1, genotype, take all of them and put them and separate them. Take that, put it. Take that and keep them close. Okay, it mustn't look like a gamete. And then you get a mark. And now you describe them. Okay, there's a person with a blood group AB. There's a now phenotype, you can say blood group AB. That's B. Can you see that? Although that is heterozygous B and homozygous B, but it's dominant. So that's B and that's A. And then you describe them and you get your marks. This is the last part is the pedigree diagram. What's the pedigree diagram? It shows you the inheritance of genes over many generations. The inheritance of genes over generations. It's called a pedigree diagram. It's not a phylogenetic tree. The pedigree diagram. Phylogenetic tree, it's not a phylogenetic tree, it's the inheritance of genes over generations. Squares represent male, grandfather, son, father-in-law. Circles represent daughters, mothers, grandmothers, daughter-in-law. Okay? So, any square. So, if you see Pelusa, you don't know now. Is Pelusa a female or male? But, oh, it's a male. It's a square. Okay, Anju. Oh, Anju. It's a female because it's a circle. Gabby is a female. Pete is a male. So you can see. And then here they're showing you that Anju and Pelisa have 4 children, 2 boys and 2 girls. Okay. So how many generations are shown here? 1, 2, 3. It's an inheritance of genes over 3 generations. Where this is P1. That's the mother and father. Then they have their children. And then these children mate and they have that children. So this is P1, that is F1. When the F1 mate, then the F1 becomes what? P2 and this becomes F2. So this is the second generation of children. So that's the first generation of children. That will be the second generation of children, but there's 3 generations. So name the type of diagram shown. It's a pedigree diagram. How many females are in this family? Count the circles. 1, 2, 1, 2, 3. Whether it's a shaded circle or whether it's an unshaded circle. If it's a circle, it's a female. And don't forget, they also put that there. That's a female. They never put circles because they were running short of space. But the female says 1, 2, 3, 4, 5, 6. How many females? There's 6. How many males in the F1 have Gall syndrome? They tell you Gall syndrome is a sex-linked disorder. Straight away, children, you know, for Pete, I must put XY. For Gabby, I must put XX because it's a sex linked. Anju, I must put XX. Pelusa, I must put XY. Gabby, okay, and these 2 children here is going to be XX and that's going to be XY. All right? And now you're going to see, okay? They're saying Gall syndrome is a sex-linked disorder caused by a dominant allele and they'll give you what to put. It's X because the allele is found on the X chromosome, not on the Y. G, it's caused by a dominant allele, X to the power big G. So anything with the X to the power big G means that person got Gall syndrome. And you can see he's affected. He's affected, okay? All right. He's got affected. Now let's see now. Males in the F1 generation have Gall syndrome. How many in the F1 have Gall syndrome? Only one. Only one male has Gall syndrome. There's it. He's affected. And his name is Pelusa. So that's 1. Let's go to the next one. Give Gabby's genotype. Now, Gabby is going to be what? X, X, and she is going to be affected. Ne? So she's going to be X to the power big G. But Pete and Gabby married and their children she has an affected son and an unaffected son, an affected daughter and an unaffected daughter. So she can't be affected. She's affected, yes. She has X to the power, but she cannot be homozygous affected. She has to be heterozygous. She must carry the normal. She got Gall syndrome and she must carry a normal. If you either have the disorder, you're affected, or you must be unaffected, means normal. So for normal, it means if X to the power big G is Gall syndrome, that means X to the power small g is going to be normal. So she must be heterozygous. So give Gabby's genotype. It's going to be X to the power big G, X to the power small g, because for her to be affected, she's the mother, she is going to pass her X chromosome to her sons and to her sons and her daughters. Né? So we know that the son is going to he's affected. He's affected. He's a son. But he can only get the Y chromosome from the father, which has no alleles. So where is his X chromosome going to come from? The mother. So he's going to inherit that X to the power big G. And the Y comes from the father. Can you see Pelusa? So Pelusa is going to be X, Y. Y is coming from Pete, the father. And because he's affected, he can only come from this big X to the power big G from the mother. Right. So they have 4 children. Give the phenotypes of their son. Now, always remember, children, that the father passes the X allele to the daughter, né? And the Y to the son. So if the father is affected, right, the daughter will be affected. If the father is affected, that means he only got one allele that making him affect. The father will pass his affected allele to his daughter. So here you can see now the mother is affected here, right? Anju and Pelusa. Now the mother is affected, okay? And here's the father. There's the mother. This mother here, Anju, she's not affected. She's unaffected. Can you see that? So she's XX, but she's unaffected, which means that she's going to be having a big G, right? She can have a big G and a big G and a small G, right? So let's see now. These 2 children, the Pelusa is the father. So this boy is going to get the Y. He's going to be unaffected. He's going to get the Y from the father. And then the male is going to get what? He's going to get the X chromosome from the mother. And that's going to be X to the power big G. And he's going to get the chromosome. So she's unaffected. Okay, they tell you here. Okay, they tell you Anju and Pelusa have 4 children. Give the phenotypes of their sons. The phenotype of the sons are going to be unaffected. They're going to be unaffected because the father is affected. He's X to the power small gy. So sorry, big gy, né? So he's going to pass only his Y allele to the son. But because the mother is unaffected, the mother Anju, she's unaffected, big G. So they are going to get that, let me see, no, small g. Now it's going to be small g. So small g. So children, I'm going to just write it here. For those 2 males, let's make the males here, male. The 2 sons, we're putting them there. That's the son. So the son is going to get the Y chromosome from Pelusa, which is the father. He's going to get the X chromosome from the mother. And the mother, Anju, is unaffected. So she's going to have a small g, for her to be unaffected. That means she's going to give the X to the power small g to the son, X to the power small g to the son, and X to the power small g means normal. So her 2 sons are going to be normal, right? So Pelusa is affected, X to the power big Gy. Anju is unaffected, she's normal. Here's it, right? That's what we said, she's normal. Males inherited the Y chromosome from Pelusa, so the son inherited the Y from the father, right? And the X from the mother, and the mother had the 2 unaffected alleles, and therefore the sons were unaffected. Okay, because the mother was unaffected. And please learn that biotechnology is a process whereby we use processes and organisms to manufacture products for human needs. Also genetic engineering, whereby we can improve the taste of fruits, have seedless fruits, tastier fruits, because of genetic engineering, where we manipulate genes from one organism to another. So biotechnology is where we use chemicals and processes and organisms to improve the quality of life, and where we manipulate the genes to improve the quality of fruits and organisms, which is called genetic engineering, to produce desirable traits. Also learn, children, that we have genetically modified organisms whereby the genes of certain desirable traits are transferred into an organism that becomes a transgenic organism. Stem cells are able to differentiate into any type of tissue and help in to relieve diseases like Parkinson's diseases. They can cure heart failures. They can also cure responsible for producing, for example, genetically modified organisms. They can produce bacteria to produce insulin. And please learn the steps for cloning, right? Steps for cloning means whereby you take the ovum of a female, you remove that nucleus, then you take a donor female who has desirable traits, and then you take the genes of the whole chromosome with the somatic cell, the chromosome with the diploid nucleus with all the genes, and transfer it to from the donor to the surrogate mother, and then let it divide by mitosis, and then you find you can clone a new individual with all the desirable traits. It only involves mitosis. There's no genes, there's no gametes or fertilization involved in cloning. Right. So genetically modified organisms are there to improve the lives of humans. You can produce insulin, and you see stem cells come from from our bone marrow. They are also found in the umbilical cord of a fetus. Right? And you can use stem cells to treat burn victims for spinal cord injuries, Parkinson's disease, and also cure a pancreas that is diabetic or creating diabetes. Cloning, you can see you must have a female sheep and a surrogate mother, and you transfer all the desirable genes in the diploid cell nucleus of the donor into the ovum of a female, and then just simply implant it into the surrogate mother, and the surrogate mother will give birth to an organism identical from which the diploid cell was removed with all the desirable traits. And cloning, you can reproduce carrots as well by asexual reproduction. You don't need males and females. And now we go to the next part of the paper 2, which will be done by my colleague Sina on DNA and meiosis. I wish you all the best, children, for paper 2 on Monday. Remember, you have tonight, Saturday, and Sunday to distribute your time wisely. Thank you. Okay, children, and don't forget that you need to go over DNA and meiosis, right? You need to look at all the question papers, because the question papers, together with the memorandum that's given to you in the QR code, will help you understand the mutations of the DNA replication, transcription. Look at the meiosis diagrams, children, the anaphase, the metaphase, how the diagrams are positioned, the labels. And that is all very important, but we've done the major part of paper 2. So you can just simply go through the QR code and you can look at the history, right? And all these things about the replication. You have worksheets as well that's given to you, right? You know all this, it's all in your SIP notes, right? And protein synthesis, transcription, translation, DNA profiling, right? These are simple things compared to the genetics, right? Look at the questions and the answers, and you will be able to understand this DNA and the meiosis diagrams very, very well. Remember, the meiosis is just the cell division responsible for variation and gametes, and you just need to know what happens in each phase. So, children, with that, I wish you all the best for paper 2, and do well and prepare well for end of the year as well. Thank you.